A revolution in a petri dish, the genesis of plant tissue culture. Plant tissue culture is a revolutionary technique that has transformed modern botany. It involves growing entire plants from single cells or small pieces of tissue in a controlled laboratory setting. Imagine a world where we can cultivate thousands of identical orchids from a single parent plant, or develop disease-resistant crops. Plant tissue culture makes these feats possible. The journey began in the early 20th century, with scientists captivated by the idea of totipotency, the inherent ability of a plant cell to regenerate into a complete organism. Early pioneers like Gottlieb Haberlandt and Roger Gautheret laid the groundwork for this groundbreaking field, conducting experiments that demonstrated the potential of cultivating plant tissues outside their natural environment. Their efforts led to the development of in vitro culture techniques, where plant cells are grown on artificial nutrient media under sterile conditions. This breakthrough paved the way for further exploration into the complexities of plant growth and development at the cellular level. The development of plant tissue culture marked a paradigm shift in botany, allowing researchers to study and manipulate plant growth with unprecedented precision. It opened up new avenues for studying plant physiology, genetics, and biochemistry, leading to groundbreaking discoveries about plant hormones, cell signaling, and developmental processes. The ability to grow plants in controlled environments also provided a powerful tool for studying plant responses to various environmental stresses, paving the way for developing climate-resilient crops. Today, plant tissue culture has become an indispensable tool in various fields, from agriculture and horticulture to pharmaceuticals and conservation. It allows us to produce disease-free plants, conserve endangered species, and develop new varieties with improved traits. The journey of plant tissue culture from a scientific curiosity to a cornerstone of modern botany is a testament to human ingenuity and our quest to understand and harness the power of nature. The science of tiny gardens, cells, nutrients, and controlled environments. At its core, plant tissue culture hinges on the principle of totipotency, the remarkable ability of a single plant cell to give rise to an entire organism. This inherent potential stems from the fact that each plant cell possesses all the genetic information necessary to develop into any cell type found in the plant. By carefully manipulating the environment and providing the right nutrients, scientists can coax these individual cells to divide, differentiate, and ultimately regenerate into complete plants. The process begins with selecting a suitable explant, a small piece of plant tissue like a leaf segment, stem tip, or root tip. The explant is then meticulously sterilized to eliminate any potential contamination from bacteria or fungi, which could outcompete the plant cells in the artificial environment. This step is crucial, as even the slightest contamination can jeopardize the entire culture. Once sterilized, the explant is placed on a nutrient-rich medium, carefully formulated to provide all the essential elements required for cell growth and development. This medium typically contains a mixture of sugars, vitamins, minerals, and plant hormones, the ratios of which are adjusted depending on the specific plant species and the desired outcome. The culture is then incubated in a controlled environment, maintained at optimal temperature, humidity, and light conditions to promote healthy growth. Within this artificial haven, the cells of the explant begin to divide and multiply, forming a mass of undifferentiated cells called a callus. By manipulating the types and concentrations of plant hormones in the medium, scientists can direct the development of this callus. For instance, a higher ratio of auxins to cytokinins promotes root formation, while the reverse encourages shoot development. This precise control over growth and development is one of the key advantages of plant tissue culture. Cloning plants, from single cells to fields of green. One of the most remarkable applications of plant tissue culture lies in its ability to create clones, genetically identical copies of a parent plant. This process, known as micropropagation, allows for the rapid multiplication of elite plant varieties, preserving desirable traits like high yield, disease resistance, or unique flower color. Imagine a rare orchid species on the brink of extinction. Micropropagation offers a lifeline, enabling the production of thousands of identical offspring from a single surviving plant. The process begins with selecting a suitable parent plant, chosen for its superior traits. A small explant, often a shoot tip or axillary bud, is carefully excised and sterilized before being placed on a nutrient-rich medium. Under the right conditions, the explant develops into a small plantlet with roots and shoots. This plantlet can then be divided and subcultured onto fresh media, creating multiple copies. This cycle of multiplication can be repeated numerous times, exponentially increasing the number of plantlets. Once the plantlets reach a suitable size, they are carefully transferred from the artificial environment of the culture vessel to a greenhouse or nursery. Here, they undergo a process of acclimatization, gradually adjusting to normal environmental conditions before being transplanted to the field. Micropropagation offers several advantages over traditional propagation methods like cuttings or grafting. It allows for rapid multiplication rates, producing thousands of plants from a single explant in a relatively short period. This is particularly beneficial for plants that are slow-growing, difficult to propagate, or commercially valuable. Furthermore, micropropagation ensures genetic uniformity, guaranteeing that all offspring inherit the desirable traits of the parent plant. This is crucial for maintaining the quality and consistency of crops, especially in the case of hybrid varieties. Section 4. Beyond multiplication, the diverse applications of plant tissue culture. While large-scale plant multiplication remains a cornerstone of plant tissue culture, its applications extend far beyond cloning. This versatile technique has found its way into various fields, from research and conservation to medicine and industry, revolutionizing our understanding of plants and their potential. In research laboratories, plant tissue culture serves as a powerful tool for studying plant physiology, genetics, and biochemistry. The ability to grow plant cells in a controlled environment allows researchers to dissect complex biological processes at the cellular and molecular levels. For instance, plant tissue culture has been instrumental in unraveling the mechanisms of plant hormone action, cell signaling pathways, and responses to environmental stresses. Furthermore, plant tissue culture provides a platform for genetic engineering, enabling the development of crops with improved traits. By introducing foreign genes into plant cells and culture, scientists can confer desirable characteristics such as resistance to pests, diseases, or herbicides. This technology has the potential to revolutionize agriculture, leading to increased food production and reduced reliance on pesticides and herbicides. In conservation efforts, plant tissue culture offers a ray of hope for endangered plant species. By preserving and propagating endangered plants in vitro, we can create a genetic safety net, safeguarding these species from extinction. Plant tissue culture also allows for the exchange of plant material between different countries and institutions without the risks associated with transporting whole plants, facilitating international collaboration in conservation efforts. Section 5. Battling diseases, one cell at a time. Plant diseases pose a significant threat to global food security, causing substantial crop losses and jeopardizing livelihoods. Plant tissue culture offers a powerful tool in the fight against these diseases, enabling the production of disease-free plants and the development of resistant varieties. One of the key applications in this realm is meristem culture, a technique that exploits the fact that plant viruses often struggle to infect the meristem, the actively growing tip of a plant shoot. By carefully excising and culturing the meristem, scientists can produce virus-free plants from infected parent material. This technique has been instrumental in eradicating systemic viruses from important crops like potatoes, sugarcane, and strawberries. Moreover, plant tissue culture plays a crucial role in screening for disease resistance. By exposing plant cells and culture to pathogens or toxins, researchers can identify individuals that exhibit resistance. These resistant cells can then be regenerated into whole plants, serving as a valuable resource for breeding programs aimed at developing disease-resistant varieties. The ability to rapidly propagate these resistant varieties through micropropagation further amplifies the impact of this technology. By producing large numbers of disease-free and resistant plants, plant tissue culture contributes to sustainable agriculture, reducing the reliance on chemical treatments and promoting food security. Section 6. The future is green plant tissue culture and the fate of global agriculture. As we face the challenges of a growing global population and a changing climate, the importance of sustainable agriculture has never been greater. Plant tissue culture, with its ability to enhance crop yield, quality, and resilience, holds immense promise for shaping the future of food production. One area where plant tissue culture is poised to make significant contributions is in the development of climate-resilient crops. By exposing plant cells and culture to various environmental stresses like drought, salinity, or extreme temperatures, researchers can identify and select for tolerant varieties. These varieties can then be further propagated and distributed to farmers, helping them adapt to the challenges of a changing climate. Furthermore, plant tissue culture can contribute to reducing the environmental footprint of agriculture. The production of disease-free plants through techniques like meristem culture can significantly reduce the need for chemical treatments, mitigating the negative impacts of pesticides and herbicides on the environment and human health. The development of genetically modified crops with improved traits like nitrogen fixation or enhanced nutrient uptake can further contribute to sustainable agriculture. These crops can potentially reduce the need for synthetic fertilizers, which are energy intensive to produce and can contribute to environmental pollution. Section 7. A new era of botanical understanding. Beyond its practical applications, plant tissue culture has ushered in a new era of botanical understanding. By providing a platform to dissect and manipulate plant growth at the cellular level, this technique has revolutionized our understanding of plant physiology, genetics, and development. The ability to grow plant cells in isolation has allowed researchers to study the intricate mechanisms of cell division, differentiation, and morphogenesis. Plant tissue culture has been instrumental in unraveling the roles of plant hormones, signaling pathways, and gene expression in these processes, providing unprecedented insights into the language of life at the cellular level. Furthermore, plant tissue culture has enabled the development of model systems for studying various aspects of plant biology. The ability to generate genetically identical plants through micropropagation has been particularly valuable in research, allowing scientists to control for genetic variation and study the effects of specific genes or environmental factors. As we delve deeper into the intricacies of plant life, plant tissue culture will continue to serve as an essential tool for scientific discovery. The knowledge gained from these studies will not only advance our understanding of plants, but also pave the way for developing innovative solutions to global challenges in agriculture, medicine, and environmental conservation.